don't think that that's true. Okay. So as we get started, I would like for you all to remind me of the 4 steps of DNA replication. What's step 1? Yeah, Emma. Thank you, Emma. Unzip the 2 strands. And Kyle, what molecular machine is responsible for unzipping the 2 strands? Helicase. How many E's are in that one? 2. Helicase. Yeah, but Kyle's version of it drew it out. There were at least 7. Okay. Step 2. What is step 2 of DNA replication? Emma. Write a short portion as RNA nucleotides, which you did last class as your blue inked letters, right? And what molecular machine does that? Lydia? Primase. Step 3. Tori.
DNA nucleotides.
Yeah, now you start adding DNA nucleotides once you've got those RNA in place. And what molecular machine does that?
DNA polymerase.
DNA polymerase 3. DNA polymerase 3. Step 4. Katie.
You take out the RNA and fix the next.
Yes, you replace the RNA nucleotides with DNA nucleotides and fix the nicks. And so that's 2 things that need to happen in step 4, requiring 2 molecular machines, and they are? Katie?
DNA polymerase 1.
DNA polymerase 1 and ligase. Excellent. Alright. So, what are the 3 big processes involved in gene expression? In the central dogma of molecular biology, there are 3 big processes. Isa.
Is it initiation, elongation?
That's the 3 steps of the first and the third process.
Oh, wow.
But the 3 processes, Mark. Transcription.
Translation. RNA modification.
Yes, so transcription, RNA modification, and then translation. Those are our 3 big processes involved in taking the DNA and getting protein. Now, the central dogma of molecular biology tells us 1 gene, 1 RNA, 1 protein. We know that's not true because the DNA oftentimes gets transcribed but never translated, right? So it'll go through transcription, it'll never go through translation, and even those that do, it's not 1 gene, 1 RNA, 1 protein. It's 1 gene, maybe 100 RNAs, and 100 different proteins. Okay? Right? Makes sense? We all okay there? Alright. So we need to talk about these in a little bit more detail, and there are 3 questions that we need to answer when we're talking about transcription. Okay, so we're going to go through transcription, RNA modification, and translation in a little bit more detail. Okay? You ready? I want you to write these questions down, and then the information that we'll come up with to address them.
Initiation, elongation.
Yes. Yes. But we're not doing that. That was 1.0. This is 2.0.
But we didn't finish 1.0.
We did. Yeah, we did.
But you didn't give us what goes after the errors.
That's what 2.0 is.
That's 2.0. Okay, so transcription. What machines are required? So when we talk about DNA replication, there are several molecular machines required to replicate the DNA. Right? Rick came up and he demonstrated 3 of those machines for us, right? You demonstrated helicase, primase, and DNA polymerase 3. Yeah. And then you all got to be helicase, primase, DNA polymerase 3, and DNA polymerase 1 and ligase. Y'all got to be those 5 molecular machines. So in transcription, we need to know what molecular machines are required. Okay? What molecular machines are required? And then we'll talk about what they actually do. First, molecular machine. RNA polymerase. So when we are replicating the DNA, we need to write a new strand of DNA. Yes? And what writes a new strand of DNA is DNA polymerase. Well, when we are transcribing, we are not writing a new strand of DNA. We are writing an RNA copy of the DNA, right? So it only makes sense that it would be written by a machine called RNA polymerase. Do you love it?
Love
You love it. Yeah. Oh, it's super cool. RNA polymerase. We also need molecular machines that are called transcription factors.
Are we gonna like write stuff like after RNA polymerase? Or like is it just
No, not right now. Right now we're just getting the molecular machines out in the open. Then we're going to talk about a second question. Then we'll talk about a third question, and the third question will just go through the overall process,
Kind of
incorporating
bothersome
some of these
that
machines.
We didn't finish this.
We did finish it.
No. Is that all that there was?
Yeah.
But I put arrows after all of them.
Well, I don't know how you did that.
I didn't put arrows on the board after all of them. Okay, so as far as molecular machines are concerned, we've got RNA polymerase, we've got transcription factors, and that's pretty much it. That's pretty much it. When we are copying the DNA, it requires us to have a molecular machine that unzips the two strands. RNA polymerase takes care of that. It requires us to have a molecular machine that writes a short RNA primer so that DNA polymerase can take over. RNA polymerase is self-priming. It can start the process. Requires us to replace RNA nucleotides with DNA nucleotides. We don't have to do this here because we're writing an RNA copy, right? It's beautiful. It's beautiful. Yep, it's beautiful. So what molecular machines are required? RNA polymerase, transcription factors. It's a pretty good answer to that question.
Next year.
Next year what?
And then everyone
We're
will
dressed up
know
as
who
you.
As me? Nobody dressed up as me, but some people dressed up as some other teachers.
No
I
one
was
knows you except for us.
a little
I
sad.
know.
Yeah, but none of you dressed up as me. There are 13 of you.
I'm not
I
wrong.
could have dressed up as Dr. Engel. Rick, you should have done it.
Why not?
Just put your hair.
Oh my gosh, you.
You might be the most qualified to dress up as Dr. Engel because you and I are both
See,
redheads.
Mr. Freeman was pretty
She
solid.
looks so much like him.
She did? She was him?
Yes.
I didn't
Okay.
know her.
He's really solid.
The next question. How do the machines know where to work? Bless you.
We always knew Kyle was free.
How do these molecular machines know where to work? How do the molecular machines know where to work? True or false? The entire set of DNA is written as RNA at some point in the cell's life.
False.
It's false. It's only how much?
3.
Not the 3.
Like 1%, right?
25.
I don't even like
I was
that.
thinking of
Just
the
think like the top 3.
Yeah,
Oh, yeah, okay. No, so I gave you the percentage. What percentage of the DNA is transcribed at some point? I gave you that number. 75%.
And 1.2 for the protein.
So 75% of the DNA is transcribed at some point of the organism's life. Okay? That's 25% of it never gets transcribed.
So is that case?
And only 1.2% of it gets transcribed for the purpose of being translated. So the question is, how do these molecular machines know where to work? It's not like when you're replicating the DNA and you have to write all of it, right? You all had that little paper chromosome on Monday, and you had to copy the whole thing. That's easy to do. You're like, we're just going to copy the whole thing. But how do you know if you don't write the whole thing? Where do you work?
I don't know.
Where do you work?
But you're gonna tell us.
I am going to tell you, but I'm going to switch to the black marker.
Yeah, we like that.
I like the color coordination.
Thanks.
Miss Burks.
Are you dressed up as Miss Burks today?
Yeah, I guess. So I didn't want to erase it because I don't know if she's covering for Dr. Phillips again today, and then it'd be messed up if I erased it and then she comes back for second period.
Yeah, I'm sure she was.
Okay.
Miss Burks sounds like a nice
She
lady.
is nice.
Okay, every gene,
Every gene.
thank you. Every gene has a promoter in front.
Oh, PYP? Oh, wow. I don't know.
Every gene has a promoter in front that basically screams. These are in quotation marks because this is obviously an what do we call this? Anthropomorphosis, but what is it as a noun? A personification. Thank you. This is a personification. So every gene has a promoter in front that screams, transcribe me. Every gene. Every gene in our genome has a promoter in front of it that basically is screaming, transcribe me. And if this is a coding gene, for coding genes, the promoter is called the Tata box. For coding genes, the promoter is this sequence: TA, TAA, TA, TAA, and it's called the Tata box or the Tata box. Emma.
What if the gene is lacking for codon?
If the gene is what?
Like, the promoter is non-existent for that gene.
Then the gene would not be transcribed.
Is that normal, or is that just like a mutation?
That would probably be one of the causes of a genetic mutation. Yeah, if you lost the promoter for a very important gene. Now, some genes are more important than others. Some genes, we call them housekeeping genes because they're necessary in order for life to be possible. Some genes are like, hey, life will work if that gene is non-functional, but you might be missing something. Like, you might not be able to deposit pigment, or you might not be able to do this with your thumb, right?
I can't do that with
So it's
Yeah, Emma.
Okay. Is there a case like the gene for eye color is just like not there?
So, eye color,
It's like
human eye color, it's like 15 or 16 different genes, but you could be missing one of them that would influence your eye color,
Could
sure.
you like be missing all of them so you have like a white eye?
Sure, but it wouldn't even take that for you to not be able to deposit pigment in your eyes.
Oh.
Yeah. Okay. So these molecular machines know where to write because there is a common sequence in front of every gene that is basically screaming, transcribe me. This is what needs to be written into RNA. Okay? Now the promoter is different for our non-coding RNAs, but we'll talk about that later. Okay? So what machines are required? RNA polymerase, transcription factors. How do the machines know where to work? There's a promoter in front of the gene that's declaring, that's screaming, transcribe me. Okay?
I never knew cells were so complicated.
I hope they were. They were simple. Then
Well, so at the time that Charles Darwin published On the Origin of Species and basically popularized an idea of descent with modification, of getting the diversity of life without a creator, they knew very little about the cells. The cell was just basically thought to be a bag of pieces and very simple. Alright, any questions on these? Because I'm going to erase this. We have one more question. Alright, I'm good. Thanks. Yeah, the Tata box, I like that.
bouncer. Hey, females, come on. Hey, you're not on the list.
Alright, last question. Last question. How does transcription work? Bless you. How does transcription actually work? How do these various molecular machines function to get us an RNA copy of the DNA? Glad you asked.
I did ask.
How does transcription work?
Yeah.
I'll tell you right now, Kyle.
Wait, I wanna know too.
If you go.
I'll tell you right now. Hey, are you allowed to wear hats in class?
I just didn't want to.
On like Spirit Week.
You can leave your hat on.
What are you supposed to eat? What is it?
Oh. Well, I just noticed like Kyle took a hat off because he's got hat hair and then you're not wearing your visor.
Yeah, it was obstructing my
Okay.
vision. It's like not have visor.
Here's how transcription works. During initiation. Initiation? Is that spelled right?
Sure.
Yeah.
Transcription factors. I need an S there. Transcription factors
That's a lot of eyes.
bind, it is a lot of eyes, bind to the promoter, I tried to skip two, to the promoter and recruit, is that spelled right? I don't know.
Yeah, yeah.
Yep.
RNA polymerase.
Free car and housing.
RNA polymerase?
RNA polymerase. So there are two molecular machines already involved in our very first part of transcription. Because transcription, step one is initiation. During initiation, transcription factors bind to the promoter and they recruit RNA polymerase. Look at that. There's step one.
Radical.
Thanks. You guys are so flattering.
Thanks.
During elongation.
Oh, there's more.
Oh, there is more.
Oh, wait.
There's always more. During elongation, RNA polymerase unzips, reads, and copies the DNA. During elongation, RNA polymerase unzips, reads, and copies the DNA. But not as DNA nucleotides, but rather as what?
RNA.
RNA nucleotides, right?
You could need N elongate, or is it elongation, or
Oh my goodness.
that's sad coming from me because I can't spell.
But no, you know what's really sad is the only interaction I'm actually getting from you all today is Dr. Eagle, I think you missed the letter.
It's okay. It happens.
And then just fake flattery. Like, oh Dr. Eagle, we love you.
We're not
How is this fake?
Oh Dr. Eagle, you're the best.
You are.
I mean, it's true.
He's always blushing. Coach Red.
And then finally, during termination,
I mean, so am I. I can't say anything about that. Ow. I'm just so flattered all the time.
everything falls apart.
Splash.
No, that was darker. It's not like
It's like a collar.
During termination, everything falls apart. RNA is released,
Well, you'd be writing like an essay.
and DNA goes back to normal. You guys are terrible.
We know.
If
I don't even-
dressed up as you today, would you still be saying that we were terrible?
I think we got one. No, we all know that.
You know what? Yes, unless all 12 of you dressed up as me.
Dude, we should have
Next
done
year.
that.
Next year.
Next year,
Everyone's gonna be in the same class.
every day, every day, everyone being cool.
I hate tomorrow's day.
Oh yeah,
Tomorrow.
tomorrow.
Cowboys
Cowboys.
versus aliens.
Yo, whose house
You
chose
just wear
that?
flannels.
Whose house chose
Idiots.
that?
They didn't ask us.
They didn't even ask us.
I think it was only the captains and silly.
Now, if you could do both, like you're dressed up as a cowboy and have an alien mask, that would be
An
respect.
alien with a cowboy hat.
An alien with a cowboy hat.
I'm alien.
I'm gonna just bust out a cowboy hat.
What about if you're just an alien that walks like they've been riding a horse all day?
Oh my god.
Really?
Johnny's bowling is so bad.
Yeah, dude, Johnny walks like he's been riding a horse
Johnny's
all day.
bowling bad.
Okay,
What?
So
Legs are like this. Johnny looked for hips.
When Lucas
Kate,
does a run.
so does this make sense?
Yes.
Do you understand what's going on during transcription? So at the end of transcription, what do we have? After transcription has happened, the DNA goes back to normal and it returns to its native state,
RNA.
and we have an RNA copy of it.
Whoa.
Whoa! And that RNA copy can go through the next part, which, Mark, what is the second process of gene expression after transcription?
Translation.
Yeah, but in between the two, because I wrote it in between.
RNA
modification. All right. Any questions?
No, sir.
Any questions about this? You're doing great. Everybody's wonderful.
Always.
Do you dye your hair?
Do I dye my hair?
Yeah, are you a fake ginger?
I have a comment about that.
A comment about being a fake ginger?
Yeah. When I was fake ginger. When
Guys,
I
I
was
confession.
2 months old, my mom was at a park and a lady asked her if she had dyed my hair, and my mom's like, "She's an infant. Like, what do you mean?" And then the lady's like, "I don't know.
Do I look like some kind of freak to
My mom's
you?
like, "That's terrible."
Yeah, you know what? I totally did, because I wanted my 2-month-old baby to be a redhead.
I think that's what she actually said.
Yeah, so I just dye it every day.
Yeah.
I don't want any of those roots to show. We gotta get this thing every day. Alright. No questions?
Stay committed. Committed to
Alright.
the lifestyle.
Are either of your parents redheads?
No, and none of my grandparents.
Neither of her
She's
brothers.
unique.
And none of my brothers.
So you need to tell your mom to request my son
Oh yeah.
as a student. I don't know if she's allowed to request students.
Are your kids coming next year?
Yeah. And my son will be in kindergarten.
And I watched the Reddit.
What about your daughter?
She'll be in 3rd grade. My kids are just gonna take this school by storm. You know?
Are you guys leaving?
Yeah.
You are?
Yeah. Why do your parents want to let us live in your house and you guys can move somewhere else?
You can totally can.
And preferably for free, you know? All right.
Oh, it's crazy. You are too.
Hey, all. This can't be the new normal, because
It's Tori.
the level of background noise.
You want me to do it? I'll do it.
The level of background noise has been increasing, and we're at a place right now. It's hard. It's hard to function. It's hard to think. It's hard to think. And then, so what's happening, I just want you to know, I want you to know what's happening inside my mind right now. I'm trying to be involved in 4 different conversations while also trying to teach you. And I feel like I might have a psychotic break.
So like maybe just limit it down to 3 conversations.
Or to none.
Wait, so
Yeah.
instead of transcription, we're now on RNA.
Yes, we've left transcription and we're going to RNA modification.
Gotcha.
Yeah, Carrie.
Is RNA modification in between transcription and translation?
Yeah, it sure is. Which is why hopefully it ended up that way in your notes.
Yeah, but I couldn't remember if I did that on purpose or not.
Oh yeah, you did. I totally did on purpose. Okay, so RNA modification. This is a process you do not find in prokaryotes. And somebody remind me what a prokaryote is. I know it's been a little while since you did that little creative writing thing.
Is the cell?
It is a cell, Katie.
It's bacteria.
Bacteria, archaeans, these are cells that lack what major structure? The nucleus and all membrane-bound organelles. Okay, so prokaryotes, their DNA is not packaged separate from everything else. They transcribe it as it's being transcribed. It's being translated. There's no RNA modification. This is only in eukaryotes. Okay, and somebody remind me the 3 steps of RNA modification that we talked about on Monday. Karen. Capping.
Polyadenylation.
Polyadenylation.
And alternative splicing.
And alternative splicing.
Wait, is today Wednesday?
I like the word splice.
It's a good word.
Constantly.
It's a good word. What's my favorite word?
Now, comma.
Last class you asked me something else. What was my favorite? I don't know.
Punctuation.
Punctuation. That wasn't last class. That was several weeks ago. My favorite word.
That was last class.
That was it?
I asked you if you would only write capital or lowercase letters one time.
My favorite word in the English language is sphygmomanometer. Sphygmomanometer. It's the machine that you use to read blood pressure.
Mine is.
Mine is isopropyl alcohol.
Okay. So step one. So step one is capping. And in capping, you add a guanine nucleotide. And why do we call it a guanine nucleotide? Somebody remind me. Because the base is G. Okay? So you add a guanine nucleotide backwards at the 5' end. And somebody remind me, please, what the 5' end means. Katie?
Well, the strands are oriented 5' to.
They sure are. They are oriented 5' to 3'. DNA polymerase writes 5' to 3'. So 5' would be the beginning of the strand. It's the beginning of the transcript. Just like DNA polymerase, RNA polymerase also writes 5' to 3'. So this is at the beginning. So add a guanine nucleotide backwards at the 5' end. And this is the beginning. That's a very important piece of information.
Some nugget.
That's a little nugget of gold.
Chicken nuggets are so good.
Okay, so that's capping.
Look what you did to him.
You know, I feel sometimes like teaching this class is like taking the dog from Up and trying to get it to just focus on something
There are like 12.
for like 5 seconds.
It's these ones.
These ones?
That one.
That one.
Okay.
1, 2, 3.
Polyadenylation. Add 50 to 200 adenine nucleotides. And why do we call them adenine nucleotides? Somebody remind me.
A base.
Because they have A as the base. Adenine nucleotides at the 3' end. And if the 5' end is the beginning, what is the 3' end? It's the end! It's the end! Whoa! Whoa! So your polyadenylation, you add 50 to 200 adenine nucleotides at the 3' end. Now I told you on Monday, these two are required in order to get RNA to leave the nucleus. If you don't do these two, RNA will not leave the nucleus. This allows it to basically have a signal that tells the cell to transport it out of the nucleus. This protects it in the cytosol. Now you eat living organisms, yes? Whether you're eating plant material or animal material or fungal material, like you're getting down on some mushrooms, right? But those are living organisms. Now they're probably not alive anymore at the time that you eat them. But they used to be living organisms, and living organisms have DNA. Yes?
Even if they're like dead and cooked.
Oh yeah.
Intaking DNA.
And so you're intaking DNA and your body uses it as an energy source.
Whoa.
So your body inside of your cytosol, inside of your cytosol, your body produces enzymes. Your cell has enzymes to break down nucleic acids and to harness energy from them. And so once the RNA leaves the nucleus, it's now exposed to those enzymes that eat nucleic acids. And RNA is a nucleic acid, is it not?
Sure.
It sure is. And so these 50 to 200 adenine nucleotides at the end, they function as a place that can be eaten while it's being translated, and it doesn't make a difference because it doesn't get translated anyways. It's like if you're trying to like get into your house and you've got a dog that belongs to your sibling that hates everybody but your sibling. Do any of you have a house like that? That was my reality growing up. My sister had a dog and his name was Bruiser. And this dog hated everybody.
Son Bruiser.
This dog hated everybody except my sister. And so anybody walked into the house and the dog started attacking them.
What?
I mean, it's no joke.
Why did you keep that thing?
We didn't for long. My dad told my sister, you either need to move out. My sister was like 20 at the time. He's like, you either need to move out and take the dog, or I'm going to put the dog down. Well, I was going to take it to the pound and eventually they'll put it down because nobody's going to adopt this dog.
No one wants Bruiser.
And my sister didn't move out, so, you know, you could maybe infer from that what happened to them.
What kind of
Anyways,
dog?
so when you entered into the house, you had to have something to give to the dog to chew on so you could make it to wherever you were going.
That's like my grandma. How many people died in this? Attacked my brother.
Nobody died. Nobody died. So
Wait,
this is the RNA equivalent to like a pillow that I would walk into my house with and hand to the dog to chew on rather than me. And so this poly A tail, this string of adenine nucleotides at the end, is something for those nucleases, those enzymes that chew up nucleic acids. It's something for them to chew on while the RNA gets translated. And the more you add, the longer that RNA survives. Does that make sense? If you only add 50, it doesn't survive as long as if you add 200 adenine nucleotides. And so the more product you need to make, the longer the poly A tail is. Emma.
Oh, yeah, I don't know. Okay. So because you can have 50 to 200 adenine
Yep.
nucleotides, is that because like does it depend on the length of the strand or like? How
It depends on how long that transcript needs to survive. If it's a transcript, like so some transcripts, some genes, the cell only needs to make a handful of proteins from that. But some of these, the cell needs to make thousands of copies of that protein, and so you need the RNA transcript to stick around for a while.
Okay.
So then you add a longer tail.
Is that because it shortens every time it copies, so you need a longer?
Well, as it's being translated, nucleases, enzymes are chewing up your RNA. And so the poly A tail is something for them to chew on before it starts making a difference.
What kind of dog was Bruiser?
He was a mutt. Yeah.
Was he big?
He was pretty big. Yeah, he was like a 65-pound dog. Okay, and what is step 3? And I don't know, maybe somebody adopted Bruiser. And they had a relationship like my sister had with Bruiser. My sister and Bruiser, they were good. They were on good terms. Everyone else with Bruiser, they were not on good
Did he
terms.
go just young?
Yeah, a puppy.
Oh, that's probably why.
Okay, so what is step 3 of RNA modification?
It's
Alternative splicing. Oh my goodness. Alternative splicing. And I told you on Monday, the last time we had class, that these two are required to get the RNA to leave the nucleus. This one actually adds to the diversity. It makes it to where from one RNA product, one RNA transcript, you could get dozens or even hundreds of different RNAs. Right? Do you remember that?
I remember that.
Okay. Any questions so far? I feel like you all have a lot of questions for each other, but not so many for me, Tori.
Okay, so if the capping is the beginning of the polydenylation.
Well, all three of these are at the beginning of the strand, sure.
Yeah, like I know you said the alternative splicing adds to it.
Yes, adds to the diversity.
Yeah, so does it add like to the beginning or to the end or like?
I'll show you right now. Yeah, we're gonna explain what's going on during alternative splicing.
Okay.
All right. And I'm gonna describe RNAs or alternative splicing by showing you what it looks like. Okay. And
So
so
you're gonna draw a picture?
I'm actually gonna project something.
Oh.
I kind of want to
Do you also draw what you're projecting?
You know what? I'll draw it. Let's draw it. Okay.
We have a paintbrush if you want.
Just paint on the whiteboard. Okay, so check this out. Here's the 5' end, which is the beginning. Here's the 3' end over here. Check this out. There's the end.
Wow.
That's part 1 of your drawing.
Yeah.
And now we've got to draw this RNA transcript So that line right there, going from the 5' end to the 3' end, that's our RNA transcript. And then over at this 5' end, what's there now? What did we do at the 5' end?
We added a guanine.
We added a backwards G. Look at that. We added a guanine nucleotide backwards. Yeah.
What's the significance of it being backwards?
It's unique and unusual, and it's a signal to the molecular machines to shuttle it out of the nucleus. Yeah. Right? A backwards G. This is unique and unusual. And then what do we do at this end, everyone?
You add 50 to 200 added nucleotides. Times 17.
Going for pity.
No what the single.
Double edge. Hello?
Okay. So far, this is what we've done to modify this RNA. We put a backwards guanine nucleotide at the beginning. We put a whole bunch of A's at the end. Now we need to do alternative splicing. And so alternative splicing, if we've got these regions, so these bars don't exist. These bars I'm putting up as divisions to show you various regions on this. This is not actual morphology on the RNA. It's something I'm adding, right? I don't know if you knew this or not, but when you drive across the country, you don't actually drive across like a physical line drawn on the ground that you move from one state to another.
Shock.
That'd be so cool.
It'd be helpful. I mean, there's usually a sign that says like "Welcome to Texas," and then some states are really obnoxious about it. It seems to be the states that nobody really wants to visit. They are really obnoxious about welcoming you to the state. New Mexico, Ohio. Ohio's really bad. You know about Ohio, right?
What do you mean
visited
obnoxious?
When you drive into, so you know, have any of you driven into Arizona on Interstate 10?
Yeah.
Like, and you cross the Colorado River, and there's like a little sign, and it's got the sun on it and says "Welcome to Arizona." And you're like, cool. Like, I feel welcome. That's very nice of you. When you drive into Ohio, you drive through a tunnel. Like it's not really. It's like when you drive and you're about to do like a big freeway interchange in Southern California. And you drive over like a big sign that has all these various signs showing you which lane you should be in. But the entire thing just says "Welcome to Ohio. Thanks for visiting." And you're like, wow, wow. If your state were a little bit better, I might want to stay. But I do feel very welcome.
The Texas sign is cool.
The Texas sign. Yeah, it's. Anyways, state pride is a very interesting thing. Okay. So all that to say, these lines, these are not actual features on the mRNA. These are things that I'm going to show you because I'm going to break this up into regions. Mark.
Kind of going back to the topic of states, which is a tiny bit. Like I remember, hearing from other people that outside the state.
Yeah.
That basically everyone hates California except for California.
I don't. Yeah, I think it's everybody's just kind of jealous of California because like the weather's really nice. And like you could go to the beach and the mountain and the desert to go like in a dune buggy all in the same day. So it's just I think it's kind of jealousy, but yeah, most states look down upon California and Californians. Okay, so what I did here is I showed you 12 regions on this RNA. And again, these are not actual morphologies, these are 12 regions. Because in alternative splicing, what we're going to do is start to cut some of these out. Trinity? No. I mean, if you want to draw 7 or 3. No, don't draw 3. That's not enough. Yeah, 3. Okay. So in alternative splicing, check this out. So in alternative splicing, after this RNA has been written, maybe these two will get cut out.
Oh shoot.
Maybe you'll cut out 2 and 6, and you'll leave the rest.
We're only cutting out 2 though?
I mean, you could cut out more than 2. In this example, I'm only cutting out 2.
How do they decide which ones to cut out?
Is it random?
It's not random.
What's the effect?
How?
We don't. It's complicated. It's complicated.
Is that okay?
It's not complicated.
Okay, and so the molecular machine that does this cut, do you see I did a cut here?
Yeah.
Like this is a universal sign for cut.
Yes.
Okay, that machine that cuts that is called the spliceosome.
Ooh.
Spliceosome?
Splice.
That's like a roller coaster.
How do I spell this? Splice.
S-O-S-O-M-E, like
Spliceosome.
Like frozen.
Splice.
Miles Frozen.
You could look it up,
Frozen.
Should have been frozen for
but that looks pretty cool.
That's splicosome.
Splice.
They're not taxable.
But he's not taxable.
Yeah.
Because no one knows what it is.
S-E-S-O-M-E. No, you need.
How do you spell spliceosome?
You need one more S-O-S-O-M-E.
Oh, I just
This is why we wait until he finishes.
spliceosome. I was just missing an O. Spliceosome.
Splice of, like the CE is the S.
I don't love
I
that,
know
but
how.
Okay, and so you've got a molecular machine called the spliceosome that will go in, it'll cut out sections of this RNA and then splice all the pieces back together. Yeah, Trinity.
What is happening between the C and the S and splice?
It's an E and an O. Is that better? Spliceosome. Okay, so that's one option. But the spliceosome could have cut out something different.
Could have.
Instead of cutting 2 and 6,
It could have cut.
maybe it did this instead. Or maybe it cut out 8 and 9.
how many can it cut out?
It can cut out all of them, but then you wouldn't have any information left.
Why do they cut out the information?
Is it like present on another?
Because it changes what this code is. Because remember, this is a RNA copy of the DNA. So if you start cutting out various sections of this,
Will it like slide
it
down?
changes. Yeah, it cuts it out and then it splices the two pieces back together.
Why is it necessary to do that?
It's necessary because it adds to the amount of diversity. Otherwise, you would need one gene. You'd need one section of DNA for every protein that the cell makes. And we talked about this last time on Monday. How many cells do humans, or how many proteins do humans make? Yeah, Tori.
Oh.
It's about 2 million.
Yeah, 1.2% of the.
Yeah, well, that's what is actually how much of it codes for protein. But human cells make 2 million different proteins. And if you couldn't splice this RNA in different ways, you would need 2 million protein coding genes. But how many protein coding genes do we actually have? About 21,000. So you have about 100 times less genes than what you would need if you could not cut this in different ways. So cutting out different portions allows you to create different products from the same gene. Keeps our genome small. Otherwise you need to pack way more DNA in. And as it is, how much DNA did we already pack into our cells?
A lot.
6 feet.
6 feet in every cell. And if you needed 100 times more coding genes, you'd have a much bigger genome. Okay? So this is alternative splicing. So if you were asked the question, let's say hypothetically, there were a question on the AP exam that asked you to explain alternative splicing, how would you explain it?
It creates variation.
You could explain it based on its outcome, the effect of it, that it increases the variation, right? It makes it to where a single protein coding gene can produce several different RNAs.
It modifies RNA.
Or you could talk about it, basically the nuts and bolts of it, right? That it modifies RNA by cutting out various portions. And these portions that are cut out, so you see there's an X there,
Yeah.
right? You see there's an X there? So this portion right here, when it gets cut out, and this portion right here, when it gets cut out, we call those introns. Introns. The sections that got cut out, we call them introns.
Okay.
The sections that remain, These sections that remain, sorry I'm writing through my spliceosome. That's terrible, but I'm doing it anyways. You see these? You see now we're referencing the sections that stayed? Do you see that?
Yeah.
The sections that got cut out, what do we call them?
Introns.
Introns, the sections that stay, we call them exons.
I was close.
The sections that got cut out, we call them introns. The sections that remain, we call them exons.
Those are really cool words.
That's
I think so too.
And it's like opposite of left.
It comes from like, I think it comes from the root of express, like what actually gets expressed. Alright?
Intron is like,
In.
I don't know what that word means.
That's pretty good.
I'm smart, but I'm not.
Any questions on RNA modification? We'll take a little bit of a break here.
I need to move
Unless
my
y'all have
legs.
some
I need to
questions.
move my legs.
Me too.
They will
Why?
cramp.
Alright.
I'll fucking kill you this morning. You got trombone?
Yeah, trombone, trumpet.
All the ones?
I love this. Is it just one?
Like horns that go on your head?
Why?
Oh my gosh.
Okay, so far, in the area of gene expression, we've talked about transcription, taking the DNA and writing an RNA copy of it, and we've talked about RNA modification, taking that RNA and in a eukaryote, preparing it to leave the nucleus, and finding ways to make it more than just one RNA product from any stretch of DNA, right? But now we need to talk about translation. And whether you were talking about a prokaryotic cell or a eukaryotic cell, translation works the same way. And there are 3 steps of translation. Somebody remind me those 3 steps. Initiation, elongation, termination. So here's the first part of the initiation of translation. Notice some of the molecular machines involved. So here we haven't even shown our RNA product yet, right? That's not even on here. So we've got a ribosome. You see this? The small ribosomal subunit. We have tRNA, a different type of RNA. Okay, both that are involved in this process of initiation. So now here's initiation step 2.
Whoa.
And all of this is being recorded. If you're like, what did we write down? I mean, I guess you can go back to where you have initiation, elongation, and termination if you want, and you can start to add some of the features.
How does this work?
How does what work?
Looks like a machine.
It won't fit all the things. I'm gonna move now. Well, yeah, but
So here's the 5' cap, that backwards guanine that we added here. They didn't draw it backwards because they're not thinking. It's attached backwards. They should do it. All the way here at this 3' end, what would you find there? A ton of adenine nucleotides, what we call the poly A tail. And here's the ribosome. Here's the RNA that we modified. We wrote during transcription, and then we modified during RNA modification. This green one. And then here's tRNA, a different one. And notice it's scanning. It's scanning that mRNA for something. It's scanning this mRNA for something. And remember, what do we call this process? We call this process translation. And somebody define translation for me.
Changing language into another.
To change from one language to another. Okay? Translation, to change from one language to another. In translation, we have to change from RNA into what?
DNA.
Protein.
Into protein.
Oh.
Okay.
And here's how we do this. We need to know how to translate this. Okay? And here's how we do it. We take codons and we translate them into a specific amino acid.
Is this just in general or is this under a step?
This is in general. Really, well, definitely during initiation and elongation. Termination is just the end. So in termination, you're not translating anymore, you're stopping it. Okay, but in translation, we take a codon and it gets translated into a very specific amino acid.
Like inside of RNA?
Well, so the codons are on RNA. So this right here, notice what this is illustrating you. The start codon. Codon.
So like from there on, is that why they're a different color because they're turning into amino acids?
Yes, they code for different amino acids. Absolutely. So if you looking at this and you're like, okay, here's the start codon, and it is AUG, adenine, uracil, guanine. 3 very specific bases strung together. So looking at this tells you that a codon is 3 nucleotides. Yes? A codon is 3 nucleotides. So if we're going to translate a codon, a string of 3 nucleotides, into a very specific amino acid, we need to have a way to do that. We need to have a way to do that. So we've got a codon, this language in the RNA. Every set of 3 nucleotides is a codon. We need a way to translate that into a specific amino acid. Notice what molecular machines we have involved. We've got this, which is a ribosome. We've got GTP, which we know is going to provide the energy necessary to do this. And look at this. On the previous slide, it told us that this was a specific type of RNA. And what was it? tRNA. Which stands for, you would think it stands for translate. It actually stands for transfer RNA. Because it is transferring the amino acid here to the growing polypeptide. And so we've got an amino acid there, and it is being brought, it is messenger. DM,
Yeah.
it's messenger. And then our RNA is ribosomal, because inside of the ribosomes you have
And
RNA
RNA?
as rRNA. There's rRNA inside here. Okay. So we've got a codon, a string of 3 amino acid, or a string of 3 nucleotides, and it corresponds to a very specific amino acid. Notice that this tRNA has 3 nucleotides that are complementary to the codon, right? The codon is adenine, uracil, guanine. What's the only thing that binds with adenine? It's U in RNA, it's T in DNA, but there's no T in RNA. Instead, you have U. And so here, adenine, uracil is complementary to it and forms hydrogen bonds. Here's uracil, so you need adenine. Here's guanine, it's still cytosine. And so notice the tRNA has a very specific, what we call the anti-codon.
Oh, it's like the.
Antithesis. I mean, antitype.
Yeah.
The anti-type?
Type and antitype.
Is that Latin?
No.
What is this?
Literary.
Oh,
Jesus is the antitype.
Yeah.
okay, I gotcha.
Exactly.
Okay, so here's the codon. The tRNA has the anticodon that matches. And if the tRNA has the anticodon that matches the codon, guess what? It gives its amino acid to this growing polypeptide, to this growing protein. But only if it matches. Only if it works.
If it doesn't work.
If it doesn't work, then you need a different tRNA.
Would it still keep moving along? Or would it wait?
No, it's got to wait until you get the right tRNA in there. Yep. Yeah, that's a good question. Okay? And so are there any questions about this? This is just initiation. It's just scanning. The ribosome and its tRNA are scanning for this specific codon called the start codon. Why is it called the start codon, do you think? It starts the process of translation. Every single protein starts with this codon and this amino acid, methionine. Yeah, Kyle.
Specifically AUG, or can it be any?
It's always AUG. Always AUG. AUG always starts the process of translation. Alright, now we need to play a little bit of a game. It's not really a game, it's more like an illumination. Okay? How many different nucleotides could you put in position 1?
For
Four.
Not for the start codon, just in general.
4.
4, why?
Because there's 4 different
There's
types.
4 different bases, right? So a nucleotide is 3 or a codon is 3 nucleotides, and so we'll go ahead and draw these 3 nucleotides. I want you to draw this out, okay? Because it gives us some information about how many codons there are going to be. Now, at this first site, we have 4 options. It could be an A, it could be a U, it could be a G, it could be a C, right? So it could be an A, it could be a U, it could be a G, or it can be a C. We have 4 options at this first position. How many options do we have at the second position?
3.
Still 4, because we can have 2 A's in a row. Yeah, why not?
It was just different on there, so I thought it was different.
Remember our Tata box? The Tata box has 2 A's in a row. The sequence that I had you copy yesterday, it had 2 T's in a row, and 2 G's in a row, and 2 C's in a row on the complementary strand.
You can't have 3 in a row?
Oh, you can have, I mean,
You can have all A's.
at the end, we've got 200 A's at the end.
True.
Yeah,
Oh, yeah.
you bet. Okay, so we have 4 different, we have 4 different bases that we can put in this position. We have 4 different bases we can put in this position. What about the third one?
4.
It's 4.
4.
So how many different codons then can we make?
16 times 4.
16 times 4, which is 64. So we can make 64 different codons. So this RNA language has 64 words in it. Okay? We've got this language. Welcome back. We've got this RNA language that has 64 different codons. 64 different codons. 64 different combinations of our bases. Does that make sense?
Yes.
Do you know how many amino acids there are in living organisms? How many different amino acids we use to make our proteins?
12.
Well, not just essential. Essential are amino acids that we can't make, so you have to have them in your diet. There are 20. 20 amino acids.
I said 18.
So there are 64 different codons, and they are translated into 20 different amino acids. 64 is greater than or less than 20, or equal to. It's greater than. So what does that tell you then? If you have 64 different codons and only 20 amino acids,
Some of the codons code for the same thing.
some codons code for the same amino acid. For instance, one of our amino acids, lysine, there are 6 different codons that all code for the same amino acid.
Wow.
Yeah. Oh yeah.
That's kind of cool.
Wow indeed.
It's like how there's different spellings of the same word.
Yes.
What?
So 64
Way.
codons get translated into 20 amino acids, plus of these 64 codons, 3 of them are stop codons, and I'll show you that in a minute. Okay? So here's initiation.
But it's the same.
No, it's like
Why
British
is it
and
really
English
just
mean
step
the same thing. Like,
Because it's
Brit
step
and
3 of
Brit, it's
initiation.
like you. But it means the same
Oh, the
word.
color?
And all of this is still pretty much in the right location.
I like the color.
We'll change this a little bit.
So that last one is initiation.
There you go.
Okay, I gotta change my arrows around a little bit, but we're pretty good. Okay, so here's the end of step 1. Step 1 is initiation. The small ribosomal subunit and tRNA scanned our mRNA until it found what? The start codon, AUG. Yeah, the one that matched it. But there's only one that'll match it, and that's AUG. Because this tRNA that's there during initiation has that anticodon, UAC, and it'll only match AUG. Yeah, here.
So there's only one start codon in the whole.
Correct. Yeah, of the 64, only one of them will begin the process of translation, and that's AUG.
What if it's not there? Is it always?
If it's not there, then it can't be translated. If there's nowhere on the RNA where you can find AUG, then you can't translate it.
So what happens?
It just, it would bind to that small ribosomal subunit. Shh. Emma asked a really good question. Y'all are missing it.
What was the question, Alex?
Usually tend to tune you out when you ask questions.
It would bind the small ribosomal subunit, and the tRNA would bind to the mRNA, would scan it, and they would not ever find it. They'd go all the way to the end, and they'd just fall fall apart.
Wait, does it always have to be AUG?
It always has to be AUG to start the process.
So not just in this example?
Not just in this example, it's always. AUG is always the start codon. Okay. So now, at the very end of initiation, the ribosome is fully formed. We've got both parts, the small and the large. You saw in the previous slide, it called this the small ribosomal subunit, which indicates that there's a large one, right? Otherwise, why would we use small? So we've got the small, we've got the large, and we have our first tRNA translating our very first codon. And so our first amino acid is methionine, MET. So first amino acid. And now initiation is done. And then what step takes over? Once initiation is done. Move to elongation. Carry.
So if the start codon's always AUG and it's always going to have to match the same thing,
Yep.
wouldn't you always get the same amino acid?
At the beginning of every protein. Yep, at the beginning of every protein, you
so
have
why would you get
methionine.
different ones if you have the same start?
Because the second codon can be different. Or the third one can be different.
Oh, but the start one always has.
Yeah, it will always start with the same codon, but the second codon could be any of the 64 codons.
And then
Now this one, the start codon always has to be AUG. AUG is our start codon, but the second one could be anything.
so would the anti-codon change depending on the next
Yes.
codon?
Yep.
Okay.
Not for the start codon, but
Right,
for the
for
next
that
one.
one.
Yes, absolutely. Alright, are there any questions about initiation? So just like initiation of transcription, you have to get all the machinery in place. In initiation of translation, you have to get all the machinery in place. You've got to get mRNA, you've got to get tRNA, you've got to get your ribosome. All the machinery is in place. Okay? Questions? May I erase this?
Yes.
Okay. That's just initiation. Because after initiation, we basically have a protein that's one amino acid long. And it's actually not technically a protein until it gets folded correctly, but... Oh no! Anyways.
No, it's okay, Dr.
Dr. Phillips is here, you're right. I didn't want to erase the sub's name because I didn't know she was coming back again today.
Oh.
And then I just accidentally erased her name, but Tori reminded me that you're here. Okay, so here's...
Back to Dr. Phillips.
So here's elongation. So here's the start codon. Do you see that there? I know it's small, but do you see the AUG there? That's the start codon. Once this process starts, now elongation will take over. And so now you go to the next codon. You find the tRNA that has the right anticodon for it, and if the tRNA fits, it'll transfer its amino acid. And then you just keep growing this by having more and more tRNAs come in as you scan the mRNA. Carry and then Tori.
Okay, so after the start codon, when you go on to the next one, does it always go to the one adjacent to it, or does
Yes.
it have to find...
No, it can't skip. Once you start, you've got to read every 3 nucleotides.
What about like,
stop?
It stops when you get to a stop codon.
what if it doesn't match up with the thing?
If it doesn't match up with what thing?
The mRNA. What if the
Oh, no matter what it is, there's always going to be a tRNA with the anticodon for it.
okay.
It doesn't matter what the string of 3 nucleotides is. We have 64 different codons that we can make. We have tRNAs that can read 61 of those. There are 3 of them it can't read, and that's because there's something else that reads it. But we're not there yet. Carrie and then Tori.
Oh my gee, she put it down too. So cute.
Okay, Tori, then Carrie, you don't have your question anymore?
Oh, I thought Tori was asking.
Tori.
I have another question. So Tori.
I never asked you your question, did I? I'm so sorry, Tori.
Okay. So I understand like the whole process and stuff of like the adding the like codons and how it like translates to like the amino acids and that like changes and stuff.
Yeah.
But what does that do for like why does that happen? And like what does it do for the
It allows you to make proteins. It allows you to take, basically DNA in this case is functioning as a
so
blueprint
this is like
for
how
some
translation
machine.
works.
Yes.
Oh,
So, you need to remember that all of the work of a cell is done by what?
Proteins.
Proteins. All of the work of the cell is done by proteins, right? You all did this creative cell assignment.
So funny.
It's all about the proteins. The proteins do everything. What do cells need to do? Give me some things that cells need to do. They need to replicate themselves. They need to replicate DNA. They certainly need to stay alive, and in order to do that, they need to communicate. They need to metabolize. They need to take things in, break them down, release the energy. They need to build other things. All of that work is done by proteins. All of that work. But the proteins, the blueprints for those proteins are the DNA. So the DNA tells the cell how to make proteins. And the way that works is the DNA gets copied as a form of RNA that can leave the nucleus. The DNA needs to stay there where it's safe and protected. The RNA can leave the nucleus, giving the blueprint for building the protein. And so once you start translating it, which it always starts at this specific codon, AUG, but once you start, you read every 3 nucleotides until you get to a very special codon. But elongation is going to continue. Every one of these codons, there's going to be a tRNA that will match it, that will have the anticodon. And when it comes in, this tRNA here, it came in, it matched it. It will transfer its amino acid to this polypeptide chain, and then the tRNA will leave, go on its way. And another one will come in, opening up now another site where tRNA will come in that has the anticodon, will give its amino acid, and this will keep going until you get to one of 3 very specific codons. Carry and then Emma.
So there are 64 different tRNA, right?
There aren't.
So some of them.
Because there are 3 codons that there's no tRNA for.
So 61.
But you would think that there would be 61, but there aren't 61 either. There are 49.
So some of them code for the same.
Yeah, and it gets a little bit complicated because, yeah, that's something that you don't need to know yet, and you won't need to know unless you go into like molecular biology at university. But there are actually only 49, and that's because this third position, there's a little bit of flexibility at the third one there. It's called the wobble position, but that's beyond what you need to know.
So if the wrong tRNA comes in,
And then it would be translated incorrectly. If the wrong tRNA comes in, it can actually give its amino acid. Most of the time, it just wouldn't work because it wouldn't actually be able to bind here. Right. Notice the anticodon has to match this perfectly because it has to form hydrogen bonds. Mm-hmm. And if it doesn't, then it won't be positioned correctly to transfer its amino acid.
So then would it just leave and like keep
Yep.
trying different tRNA?
Mm-hmm. Yep, absolutely. Emma.
So can you just quickly explain what happens between the first and the second steps?
The first and the second step. So here,
Yeah.
well, step 1 was basically an initiation. So now we've attached the large ribosomal subunit, and we've translated the first codon into an amino acid. That's what's happening during step 1. In step 2, now we're translating the second codon into an amino acid.
Thank you.
Yep. Actually, in step 2, we haven't quite transferred that yet, but we've got our second tRNA in there. And then step 3, we've transferred its amino acid to our growing polypeptide. Step 4, we eject the old one and we bring in another one. Okay. So this process will continue, reading every 3 nucleotides, translating every 3 nucleotides into a specific amino acid until you reach one of 3 codons that do not code for an amino acid. And so then we go into termination. So initiation, you're just scanning your mRNA for the start codon, right? And building your machine. Step 2, elongation, you're translating every codon, every 3 nucleotides into an amino acid. Eventually you get to a point where you reach one of 3 codons that do not code for an amino acid. We call these start codons. Stop codons. Yeah, stop codons, but it's missing some of the information.
It's not very good.
No. That should be UGA, UGA, UAA, and then the other codon, the other stop codon, is it UAC? I don't know what this third position is. I'll find that one for you. Okay, so we have 3 of our codons that do not get translated into an amino acid because there is no tRNA with the anticodon that can match it. Okay, there's no tRNA with a anticodon that can match it. Instead, we have what's called a release factor. This RF, it's a release factor that binds these 3 stop codons, and it just, the whole machine falls apart. Releases your polypeptide, releases your mRNA, the ribosome falls apart, all because there was no tRNA to match there. There's no amino acid to add to your polypeptide. Emma.
So going back to the first step, after they're turned to after it codes the amino acid, what are they turned to?
After it codes,
Every 3 amino acids.
every 3 nucleotides is translated into 1 amino acid. Yep, you bet. You bet. Okay? So there are 3 of our, how many codons again do we make? No, how many different codons? 64. 20 amino acids, 64 codons, yes? 64 codons. 61 of those get translated into an amino acid. 3 of them, there's no translation. They basically get translated into stop. Okay? So 3 of our 64 codons get translated into stop. The reason that happens is because there's no tRNA to match them. There's no tRNA to match those stop codons. And so this release factor comes in, it destabilizes the whole process, everything falls apart. Your mRNA is released, now it can be translated again, but you're gonna have to assemble a new ribosome. Your tRNA that was sitting there is released, and your polypeptide is released. Okay, to go and become a protein. To fulfill its destiny.
Oh yeah.
Oh yeah.
That's not taxable.
Alright? But you had a paintbrush in your ear, or was that Carrie's paintbrush?
We're both artists.
Yeah.
She's just
I
better.
just-
I wanted to be a stay-at-home mom, and Mr. Lee said I couldn't. And I was like, but why not? Because I feel like that's a worthy and honorable job.
It's not
and
taxable.
okay, no, you can put it on tax forms. You can write homemaker
really?
on tax forms. So it's taxable too. And he was like, no, you can't do that because you don't get paid. It's like, you're saving money in childcare, so the way you think.
A penny saved is a penny earned. You can use that to do like a geometric proof to prove that being a homemaker is actually a penny. Alright.
If we do this again next year, I'm definitely just
How many
gonna
of us are there?
I thought you were gonna dress up as me if we did this.
Oh, shoot. Well, we'll see.
I'll just have this
Okay.
at home.
I'll just write my thesis on it.
Alright.
Tell us about it.
I'm gonna do it on top of that.
So we've worked through transcription, we've worked through RNA modification, we've worked through translation, and we didn't get to chapter 18 yet.
But we've got a lot of good
But we've got like 3 minutes.
so
Alright?
My muscles are about to just lock up.
you're not even using them.
That's why.
Any questions about this process? Okay, shh. How many codons are there?
49.
64.
Let's see RNA.
64 codons. How many amino acids?
20.
20. How many of our 64 codons are translated into one of our 20 amino acids?
61.
61 of them. What are the other 3 translated into?
Stop.
Stop. Okay, what is the start codon?
AUG.
AUG. So that in initiation, you're going to scan until you find AUG. And then what do you do during elongation?
You keep
You
going.
keep going.
You keep going, translating codon after codon after codon after codon. And the largest protein product in human cells is coded by, what is it? It's 100,000 nucleotides, so it'd be 30,000 amino acids. The longest protein in human cells, 30,000 amino acids. That's a lot. In this example, let me show you this example. This example, it only got to 100. You see that? Amino acid 100, and then it got to a stop codon. But the largest protein that we have is about 30,000 amino acids. Meaning during elongation, you have to read 30,000 codons before you reach a stop codon. So it can't be random. right? The language has to be very specific to be translated into a very specific string of amino acids to make a very specific protein. Carry.
Does the release factor look like a tRNA?
At one end. At this end it does because it has an anticodon, sort of. It has an anticodon, but it doesn't look like it on the other end. The end that attaches to an amino acid. So it does on one end, but not on the other. All right. So what are our three processes of gene expression? 3 processes. Transcription, RNA modification, translation. All right. Have a wonderful day.